What if we could actually bring back the woolly mammoth? You know, it's a question that feels like it's been ripped straight out of a sci-fi movie, but it is at the very heart of a real and, yeah, pretty controversial field of science called de-extinction. So today, we're going to separate the Hollywood fantasy from the incredible scientific reality. Let's get right into it.
So let's just start with that picture we all have in our heads, right? Thanks to the movies. You've got a perfectly preserved mammoth, frozen solid in ice. A scientist swoops in, pulls out some DNA with a syringe, and boom, a baby mammoth is born in a lab. Is that really how it could work? Is it even possible?
Well, here's the thing. The answer is kind of both. It's a fascinating paradox. So no, we absolutely cannot clone a mammoth the way you're thinking. But yes, we might just be able to create something new. A kind of cold-resistant elephant that looks and acts just like a mammoth. And the huge difference between those two ideas, that's where the real science begins.
To really get why, we have to start with the single most important ingredient in this whole recipe, the genetic blueprint, DNA. And here's the first big surprise for most people. DNA is incredibly fragile. The very moment an animal dies, its entire instruction manual just starts to fall apart.
Think about it like this. The moment of death is like hitting a start button on a self-destruct sequence. Those long, beautiful strands of the double helix, they get attacked by enzymes, by bacteria, by the environment itself. They just break down, shattering into millions of tiny, unreadable pieces. So that whole idea of finding a complete, intact genome, unfortunately, that's pure fantasy.
And you can see here just how many enemies DNA has. It's almost like a forensic investigation. First up, you've got temperature. Even a small rise in heat can literally double the speed of decay. Then there's moisture, which just helps chemicals break everything down and creates a perfect little home for microbes. And those microbes, they're the real wrecking crew. They release enzymes that just chew DNA to pieces.
And this brings us to a really crucial point, which is summed up perfectly by the leading paleogeneticist, Dr. Beth Shapiro. Even when we find a mammoth that looks perfectly frozen in the permafrost, the DNA inside is still shattered. There are no intact cells. There's no complete instruction manual to read from. So the Jurassic Park scenario, yeah, that's officially off the table.
Okay, so if there's no intact nucleus, then that classic Dolly the sheep style of cloning is completely impossible. That one problem forces scientists to totally abandon the idea of cloning and get much more creative. Let's do a quick refresher on how that traditional cloning even works, right? You take an egg cell, you pull out its nucleus. Then you take a nucleus from a donor cell, the one you want to clone, and you pop it into that empty egg. But the keywords there are complete, living, intact nucleus. The very thing that gets destroyed after death.
And this, right here, this is the real goal of de-extinction today. It's not about cloning, it's all about genetic engineering. So instead of trying to photocopy a shredded book, scientists are taking a brand new book, the genome of a living relative, like the Asian elephant. Then they use a tool called CRISPR, which works like a biological find and replace function, to edit in keywords and sentences from the mammoth's tattered ancient genetic code.
So how does this actually work in the real world? How do you take those ancient DNA fragments and use them to edit the genome of a living, breathing animal?
Well, the modern strategy is all about precision. The goal isn't to make a perfect mammoth copy. No, it's to take the Asian elephant's genome and basically give it a cold weather upgrade. Scientists are targeting the exact genes for the key traits. A shaggy coat for warmth, smaller ears to stop heat loss, extra layers of fat, and even specialized blood that works better in the freezing cold.
A really fantastic real-world example of this is the direwolf project by a company called Colossal Biosciences. Now, the direwolf, famous from TV and books, isn't a direct ancestor of modern wolves. So what they're doing is starting with the gray wolf genome, using ancient DNA from fossils to figure out the key genes, and then using CRISPR to edit them. The goal is to recreate traits like the direwolf's bigger size and its unique fur that was adapted for the cold. And this isn't some vague idea. It is incredibly specific. For the direwolf project, they've made 20 very targeted edits to the gray wolf's genome. Each one is designed to nudge a specific trait a little bit closer to what they believe the ancient direwolf had. This isn't a blunt instrument. It's more like genetic surgery. But this brings up a huge question about what this new animal really is. And Professor Corey Bradshaw just nails it here. Is the resulting animal really a resurrected direwolf? He argues no. It's a genetically modified gray wolf that just happens to express some direwolf traits. It's a proxy, or a hybrid, and that's a really important distinction to make. But this approach has to be adapted for different animals. It's not one size fits all.
Take the dodo, for instance. You can't just implant an embryo into a mother bird, so the whole process is different. Scientists are working with what are called primordial germ cells. These are basically the cells that become sperm and eggs. The plan is to edit the germ cells of the dodo's closest living relative, the Nicobar pigeon, and then inject them into a regular chicken embryo. If it works, that chicken will grow up and lay eggs that carry the dodo's edited DNA. Wild, right?
So all of this work, all of this incredible science, it leads us to the biggest questions of all. We've talked a lot about the how, but what about the why? Why go to all this trouble? And, maybe more importantly, should we even be doing this at all?
Well, the people who support this have some pretty compelling arguments. They believe that reintroducing huge herbivores like the mammoth could restore entire ecosystems, like turning the Arctic tundra back into carbon-trapping grasslands, which could actually help fight climate change. And beyond that, the genetic tools that are being developed for de-extinction, they could be powerful new weapons in the fight to save the species that are endangered right now from things like inbreeding or disease. On the other side of that coin, though, are some really profound ethical concerns. I mean, just because we can do something, does that really mean we should? What happens when we start playing God with the actual building blocks of life? And these questions are huge. First, what about the animal itself? What kind of life would it even have? We have no idea what health problems it might face or how a creature that was designed for the Ice Age would cope with our modern world. And then there's the ecological risk. What happens if we release a new species into an ecosystem that's already in a delicate balance? And maybe most importantly, are we just creating a being for our own curiosity, essentially defining its entire existence before it's even born?
So when you boil it all down, we're not really talking about resurrection. We're talking about authorship. For the first time in history, we have the tools to actually edit the book of life. The question isn't just can we bring back the mammoth, but what kind of mammoth should we create? As we become the authors of new species, we really have to decide what kind of story do we want to tell. Thanks for tuning in.